Hey everybody, I'm Rachel Neff, and I'm here at the Field Museum of Natural History, where I have been interning this summer in the museum's largest collection, the insect collection. The insect collection contains over 16 million specimens and features an abundance of my favorite insect group, the beetles. Beetles are the most biodiverse organisms on Earth, and they occupy countless habitats like underground soil, inside tree trunks, and even underwater. My project this summer focused on one group of these underwater beetles, the family Elmidae, or the riffle beetles. With the help of Dr. Crystal Meyer, I used riffle beetle wing structure as a proxy for dispersal ability and explored the implications this has on riffle beetle evolution. Named for the shallow, rocky portions of streams they inhabit, these aquatic beetles are found all over the world. This does not make them any ordinary beetle, though. Riffle beetles are scuba divers. They breathe underwater, never coming back up for air for the duration of their entire adult lives, which can last up to 5 years. This is possible because riffle beetles breathe using an air bubble on their bellies to take up and release oxygen and CO2 from the water around them. This need to take up oxygen from the water means that riffle beetles require clean, cold, highly oxygenated aquatic environments. Because of this, riffle beetles are useful bioindicators, meaning that their presence indicates a healthy watershed. In America, the taxonomy or classification of these useful bioindicators is well known and often utilized to assess stream health, but riffle beetles in other parts of the world are much less well studied. My project focused on a genus of riffle beetles from New Zealand called Hydora. Research suggests Hydora may be some of the most useful bioindicators in New Zealand, but scientists still don't know very much about them, specifically their evolutionary relationships. My research on this group seeks to better understand these beetles by studying their wing morphology or size and shape, because this can tell us about their evolution and speciation, or the rate of how quickly they diverge into different species. Riffle beetle speciation is strongly affected by one important life history event that relies completely upon the beetle's wings, the dispersal flight. Before new adult beetles dive underwater, they disperse by flight away from their developmental habitats, distributing the new adults throughout a watershed. The distance of this dispersal flight has major implications for speciation rates. With a shorter dispersal distance, for example, similar genes will all be concentrated in a smaller area, causing certain traits to become exaggerated, meaning new species will evolve more quickly. While it is difficult to study the dispersal flight firsthand, past research on aquatic beetles has used wing morphology as a proxy for dispersal ability. This summer, I dissected out wings from 13 Hydora morphospecies in order to analyze their wings. I dissected a total of 74 riffle beetle specimens, so about 5 to 6 specimens from each morphospecies, and I mounted their wings on slides like this. I then photographed the wings with a high-tech camera system, taking 5 to 8 pictures of each wing pair, all in slightly different levels of focus. After combining these images into one clear photograph, I then placed points called landmarks on common features of the beetle wings in order to find differences between the wings. This study will provide important information about the evolutionary relationships of these interesting beetles and will help scientists to distinguish between different species of Hydora as they are further studied.